Hereby I open this academic ceremony in which Petrick will defend the academic thesis, Nutritional Approaches to Modulate Mitochondrial Bioenergetics. May I invite you to present a summary of your study and the conclusions of your thesis.
Dear Prorector, members of the Corona, family, friends, and colleagues. In the coming 15 minutes, I will present a summary of my PhD thesis entitled Nutritional Approaches to Modulate Mitochondrial Bioenergetics. Or translated into Dutch, voedingsinterventies om mitochondriële functie te veranderen. Mitochondria are small structures that exist in almost all cells of the body. There are many mitochondria in each cell, and one of the main roles of these mitochondria is producing energy. The ability of mitochondria to produce energy is dependent on the supply of nutrients. Fats and carbohydrates are two of the main nutrients. These nutrients are transported into the mitochondria and first undergo processing. The products of this processing then lead into a series of steps within the mitochondria to produce energy. At the fourth step, oxygen is consumed in a process known as mitochondrial respiration. At the fifth step, energy is produced. This energy is then transported out of the mitochondria to be used by the rest of the cell. In addition to producing energy, mitochondria are also important for releasing signals to the rest of the cell. One of these signals are called reactive oxygen species, or ROS. ROS are produced during times of stress and can have a negative effect on the rest of the cell. Mitochondrial function varies across a range of situations. Impaired mitochondrial function can contribute to many health problems, such as obesity, cardiovascular disease, and the decline in muscle function during muscle disuse. In healthy situations, mitochondria allow us to perform physical activity and maintain daily tasks. When mitochondria function very, very well, this can allow individuals to perform a very high level of exercise. In all of these situations, nutrition has an important influence on mitochondrial function. Therefore, the aim of this dissertation was to examine the impact of nutrition on mitochondrial bioenergetics in situations of disease, health, and exercise. This dissertation was completed as a double degree between the University of Guelph in Canada and Maastricht University here in the Netherlands. This allowed us to examine mitochondrial bioenergetics in many diverse situations. We used mouse models in Guelph to study mitochondrial bioenergetics within fat tissue, skeletal muscle, and the heart. We extend these findings in Maastricht to examine mitochondrial bioenergetics within skeletal muscle of human participants. The process of measuring mitochondrial bioenergetics involves the collection of a small piece of muscle from the animal or human, an assessment of mitochondrial parameters experimentally outside the body. The two main parameters we measure are mitochondrial respiration and mitochondrial ROS. Mitochondrial respiration relates to the energy-producing ability of the mitochondria. Mitochondrial ROS are a stress signal released by the mitochondria and can have a negative effect on the rest of the cell. Together, these two parameters form our outcome of mitochondrial bioenergetics, which is the focus of this dissertation. It is known that mitochondrial dysregulation occurs during obesity. Obesity can develop from eating too much food, or alternatively, from eating food that is high in fat. However, it is not known which of these two factors causes many of the problems within mitochondria during obesity. Therefore, we aim to examine which nutritional habit causes mitochondrial dysregulation during obesity. When too much low-fat food was eaten, shown in the turquoise bars, mice gained weight and developed obesity. When too much high-fat food was eaten, shown in the blue bars, mice gained even more weight and developed extreme obesity. However, the greatest levels of mitochondrial ROS and ROS-related stress occurred only when eating too much high-fat food. This shows that many of the negative changes within mitochondria that occur during obesity are due to the high-fat components of the foods that are eaten. Since we established that eating high-fat foods can have a negative effect on mitochondria, we next aim to examine potential ways that we could prevent this. Dietary nitrate supplementation is one such nutritional approach. Nitrate can be found in foods such as beets and green leafy vegetables and has a potential to influence mitochondria. Therefore, we aim to examine if dietary nitrate supplementation would be beneficial for maintaining mitochondrial bioenergetics and overall health during the consumption of a high-fat diet. When a high-fat diet was eaten, shown in the turquoise bars, mitochondrial ROS in the heart muscle was higher compared to when a low-fat diet was eaten, shown in the white bars. In addition, heart function was lower when consuming a high-fat diet compared to when consuming a low-fat diet. However, when nitrate was supplemented with the high-fat diet, mitochondrial ROS was not higher compared to the low-fat diet. In addition, heart function was not lower. This shows that nitrate has a beneficial effect for maintaining mitochondrial bioenergetics and overall heart health during situations of obesity. Since nitrate is beneficial for improving mitochondrial bioenergetics during situations of dysregulation, such as obesity, we next aim to examine if nitrate would be beneficial in other situations of dysregulation. To do so, we studied muscle disuse. Disuse of a muscle, such as limb immobilization, rapidly leads to a decline in muscle mass, muscle function, and mitochondrial bioenergetics. We aim to examine if dietary nitrate supplementation could prevent these negative effects of immobilization. When nitrate was not consumed, mitochondrial respiration was lower in the immobilized leg, shown in the turquoise bars, compared to the non-immobilized leg, shown in the white bars. When nitrate was ingested, shown in the right panel, mitochondrial respiration was not lower in the immobilized leg compared to the non-immobilized leg. This shows that nitrate is beneficial for maintaining mitochondrial respiration during muscle disuse. In addition, mitochondrial ROS was higher in the immobilized leg compared to the non-immobilized leg when nitrate was not ingested. When nitrate was ingested, mitochondrial ROS was not higher in the immobilized leg compared to the non-immobilized leg. Combined with the beneficial effects of nitrate on mitochondrial respiration, this shows that nitrate consumption prevented mitochondrial dysregulation during muscle disuse. Given the beneficial effects of nitrate in situations of mitochondrial dysregulation, we also wanted to examine if nitrate could influence mitochondria in healthy situations when mitochondria are not dysregulated. In these healthy situations, it is known that nitrate can improve muscle performance. we aim to examine if this occurred due to changes within mitochondria. However, when healthy mice consumed nitrate, shown in the turquoise bars, mitochondrial respiration was not different compared to when healthy mice did not consume nitrate, shown in the white bars. In addition, there was no difference in mitochondrial ROS when nitrate was ingested in healthy mice compared to when nitrate was not ingested. This shows that the beneficial effects of nitrate for improving muscle performance in healthy situations are not due to changes in mitochondrial bioenergetics. Nitrate is one supplement used by athletes to improve performance. Another supplement used by athletes for potential effects on performance are ketones. Ketones are substrates like fats and carbohydrates. Similar to fats and carbohydrates, ketones are transported into the mitochondria and enter the same processing pathways. These processing pathways are what are important for producing energy within the mitochondria. Even though ketones are supplemented by athletes in hopes of improving performance, not much is known about the ability of mitochondria to use ketones. Therefore, we aim to examine if ketones are an important substrate for mitochondria. To do so, we perform muscle biopsies in trained cyclists, both at rest and following exercise. In these muscle samples, we measured the extent to which ketones could be a substrate for mitochondria. The ability of ketones to increase mitochondrial respiration, shown in the white bars, was very small. In contrast, the ability of carbohydrates to increase mitochondrial respiration, shown in the turquoise bars, was very large. This shows that ketones do not contribute much to mitochondrial energy production. In contrast, carbohydrates are an important substrate for mitochondrial energy provision. Altogether, from this dissertation, we can conclude that mitochondria are essential in situations of disease, health, and exercise. There are multiple ways in which nutrition can influence mitochondria. We show that overconsumption of high-fat foods causes mitochondrial dysregulation in situations of obesity. Supplementing with dietary nitrate can prevent this mitochondrial dysregulation during obesity, as well as during muscle disuse. During exercise, ketones are not an important substrate for skeletal muscle mitochondria, while carbohydrates are important for mitochondrial energy production. Altogether, in this dissertation, we have identified several nutritional compounds which can influence mitochondria, and this has the potential to impact human health and performance in many diverse situations. Thank you for your attention, and I give the word back to the prorector.
Thank you for your presentation. The opposition will be opened by Professor Goossens, Professor of Cardiometabolic Physiology of Obesity at this university. Professor Goossens was also the chair of the assessment committee.
Thank you, Mr. Prorector. So, dear respected candidates, I've read your thesis with a lot of interest, and I must say the cover is also very beautiful. And you performed an impressive amount of complex experimental work spreading from experiments in cells, animals to humans. And you examined the effect of different nutritional strategies on mitochondrial bioenergetics and risk factors for cardiometabolic diseases, using both classical biochemical techniques, molecular techniques, but also very interesting state-of-the-art methodologies. And I must say that the rationale behind these studies, as well as the experimental outcomes, are described in an excellent manner. And not only the quantity, but also the quality of the work you have performed is really impressive. And it's therefore not surprising, in my opinion, that almost all articles have already been published in well-respected international journals. So I would like to congratulate you, but also your supervisors, with this extensive and elegant work that you described in your thesis. But of course, I'm not here today to only give compliments. I would like to ask you some questions as well. And I would like to ask you some questions related to chapter 4. And in this chapter, you describe a series of experiments to investigate the ability of dietary nitrate to prevent high-fat diet-induced impairments in mitochondrial bioenergetics and cardiometabolic health in mice. And you showed that dietary nitrate improved lipid and glucose homeostasis, and as well it improved cardiac function. And my questions relate to the underlying mechanisms of these effects. And on page 76, you conclude that, and I quote, "It is therefore likely that dietary nitrate acts through multiple mechanisms of action, which are not solely dependent on increasing nitrate concentrations to exert a cardioprotective effect." So my first question would be, can you briefly explain what are the potential underlying mechanisms, how dietary nitrate exerts these beneficial effects based on your findings?
Dear highly esteemed opponent, thank you for your question. That was referring to our work with the gut microbiota partially, because in that study we first fed mice nitrate supplementation, and that had beneficial effects in cardiometabolic health. And then we provided feces from nitrate-fed mice to a separate subset of high-fat diet-fed mice, and that fecal transplantation also had the beneficial effects. So that shows that some of the changes in nitrate are not dependent on increasing the plasma nitrate concentrations or serum nitrate, so kind of nitric oxide independent, since we also talk about nitrate can have an effect with increasing nitric oxide causing vasodilation or other direct changes in that sense from the metabolite. But we also see beneficial effects that are not due to nitric oxide itself, so more secondary to other metabolites that are changed in a different animal.
Mm-hmm. Right, and you suggest mechanisms related to adipose tissue inflammation, gut dysbiosis, right? That might be impacted. And I would like to challenge you a little bit by stating that, at least in my opinion, the effects that you have shown are actually solely dependent on changes in the gut microbiome. So in your discussion, you mentioned that not all mechanisms, at least there might be other mechanisms involved, also the gut microbiome. But I would say it's purely due to changes in the gut microbiome. And do you have any idea why I would come up with this pretty bold statement, maybe?
It could be because a lot of the findings with the FMT model recapitulate what we saw with the original nitrate model. Mm-hmm. So the improvement in ROS in the heart, improvement in cardiac function, and improvement in serum lipids is another big factor, since lipids in circulation can have consequently influence ectopic liquid accumulation in many other tissues, and then subsequently influence metabolism.
Yeah, and can you also provide any arguments against my statement? So why would you disagree maybe that it's purely dependent on the gut microbiome changes?
I would disagree with that because of our other findings within skeletal muscle, where we see that the fecal microbial transplantation did not improve mitochondrial bioenergetics within skeletal muscle. Mm-hmm. And it did not have the same effects in the adipose tissue as well, so there is a few differences we didn't see there. So it seems like nitrate can act through multiple mechanisms, because when we just look at the gut microbiome, it didn't fully recapitulate the whole body effects that we saw of the original model.
I partly agree. So changes in adipose tissue were not seen after the FMT experiments that you did, but maybe that's not involved in the improved glucose and lipid homeostasis and cardioprotective effects. So let me maybe elaborate a little bit on why I think it might be purely due to the gut microbiome. And that is when I look at, for example, the dietary nitrate-induced improvements in cardiac function. So end-diastolic volume, stroke volume, cardiac output that are depicted on page 65, figure 4.1. And to be very specific, panels F, G, and H. There you see the parameters related to cardiovascular function. And when I compare those with the FMT results on page 72, so that is figure 4.7, panels C, D, and E. In my opinion, the effect size is completely the same, if not more pronounced with the FMT experiments. and I shall not refer to all the figures, but when I look at the data on glucose homeostasis, so area under the curve for glucose, for example, indicating glucose tolerance, and a supplementary figure 4.1, but also on in figure 4.6 in the main text, I can draw the same conclusions. The effect size is absolutely identical. So doesn't that suggest that it's purely mediated via dietary nitrate-induced effects on the gut microbiome, and that all the other mechanisms that you might see on mitochondrial loss or adipose tissue inflammation are not linked to improvements in cardiac function and glucose and lipid homeostasis?
There's like a well-known interaction between the gut-heart axis, so that's also gut-adipose-heart axis. So that's probably what we see a lot of in the gut microbiota aspect, with directly linking to the heart. But it would not be. I would also argue that it's not just the microbiome, because the effects of nitrate on increasing nitric oxide and having other effects with vasodilation or changes in that sense are also important for the whole body beneficial effects of nitrate, but it does appear that quite a lot of it is mediated through changes in the gut microbiome or release of inflammatory markers from the gut into circulation that then can crosstalk with the heart as well.
Right, maybe a very brief final question, if I'm allowed to, yeah. So you described several mechanisms, also short-chain fatty acids that are unchanged after the intervention. There could be other links, I think, with the effects you see, gut-brain axis, for example, but you don't see changes in appetite or body weight. So could there be a mechanism that you think of how these changes in gut microbiome link to the cardiometabolic benefits that you see?
So we didn't see any changes in the metabolites we measured, but it also could be release of inflammatory markers into circulation that we didn't measure. So things like IL-6 or TMAO or other kind of metabolites from the gut that weren't directly what we measured but could be involved.
Okay. I'm happy with your questions and would like to give the word back to the Prorector.
Thank you. The opposition will be continued by Professor Sprite, Professor Emeritus of Muscle Metabolism at the University of Guelph in Canada. Professor Sprite was member of the assessment committee, and we highly appreciate your participation to this academic ceremony.
Ms. Candidate, I'd also like to congratulate you on the breadth and depth of your work, many experimental models, and when you do work over a wide range of different models and experiments, it opens up a lot of possibilities. So I'm going to get right into it, and my questions are going to be somewhat philosophical. So in your presentation, you make it quite clear that ROS plays a big role in a variety of things, and twice you said that it can have negative effects on the cell. So why do you think the mitochondria is designed that way, to have this protein leakage and all this ROS production?
Dear highly esteemed opponent, thank you for your question. I would like to highlight in my wording can have negative effects, because an important role of ROS is also having adaptive responses. So ROS released from the mitochondria can increase in response to an acute bout of exercise, and we know that's important for the adaptations in response to exercise training. So it can have a negative effect in a more pathological situation, but also can have a good response to a stress signal and lead to adaptations as well.
So why do you think we're so poor at defending ourselves against a high-fat diet with respect to the ROS axiom?
I guess we always can go back to kind of like the evolution of humans to begin with, where a key part of life was eating a lot when you can eat a lot, and then exercise or run or store it when you can, use it for a time in a time of need. So in that sense, then it could be a signal to kind of initiate the storage of the nutrients when we have them available, so then they can be used at a later point during times of need. The difference now in modern societies, we are always in a surplus. So it's then a chronic, increasingly kind of perpetuating the issue of ROS emission.
So how many generations will it take before we can adapt in a favorable way to the high-fat diet?
however many generations it took us to adapt unfavorably, but in reverse, maybe a little longer because it always takes longer to recover something than make it worse.
Okay. I'm going to change tack and ask you about this somewhat quizzical response to exercise in that the mitochondrial sensitivity to ADP decreases. Why do you think that happens? There must be some reason. I know you've alluded to it in the thesis, but I didn't really see any. You might have some thoughts on why you believe this happens.
Yeah, so we know ADP within the cytosol is a potent activator of glycogen phosphorylase. So we think that the decrease in mitochondrial ADP sensitivity during exercise then promotes the accumulation of ADP within the cytosol, and then promotes glycogen breakdown to have a substrate available for mitochondrial energy production during prolonged exercise. Because at the onset of exercise, there's a rapid rise in free ADP, but then the change in ADP sensitivity could be a more prolonged mechanism that acts at extended exercise.
But do you think the magnitude of the changes that you see in your model are large enough for that hypothesis to be correct? Because it seems to me many of the times that it's measured, the magnitude of the change is not great. So I'm just wondering how that could translate into the cytosol and be significant enough to have an effect on not just phosphorylase, but all the enzymes that are activated by ADP or AMP.
In terms of the magnitude, we know there's a lot of different factors that upregulate the activity of those enzymes to begin with, so we wouldn't necessarily argue it's only due to the change in ADP sensitivity. But I think it also depends on like the localization within the cell as well. So maybe it's more like concentrating ADP in the vicinity of where glycogen phosphorylase is to promote the breakdown.
So is it possible that the sensitivity goes down so that there's a greater range over which the mitochondria might be sensitive to? In other words, if it was super sensitive all the time, then you would have a hard time sort of having a graded response to the increase.
The changes in free ADP concentrations available to the mitochondria during different intensities of exercise also do influence that. So during exercise, an intensity-dependent rise in free ADP concentrations. So in a different argument, it also could be kind of a mechanism to like modulate the amount of ADP that gets into the mitochondria. So that it's not it's not a large influx of ADP, but a mechanism to kind of promote a back inhibition.
Okay, good. Related to that, I want to ask you about the permeabilized fiber technique. And there's no question that I think it's a better technique, more sensitive technique than simply gross mitochondrial measures on isolated mitochondria. But do you have any thoughts on how physiological you think the prep is and the results that you find?
It's definitely a step more physiological than isolated mitochondria, but of course we can never model everything perfectly in vitro. So we performed the experiments within room air oxygen saturation, but that also isn't what occurs within our muscle. We titrate concentration of ADP in some maximal concentrations to look at modeling the in vivo environment, but we, of course, still use saturating pyruvate, saturating malate. So that also doesn't occur in vivo in the muscle microenvironment. So there's, of course, limitations of the method still, and it's not a direct representative situation of in vivo. So we kind of make analogies between concentrations, but there are so many other factors, blood flow, pH regulation that can change during exercise as well. So things like that we can't fully model ex vivo in our experiment.
So given that you're continuing to work in this area, have you come across any ideas for improving the preparation?
There are other ways to non-invasively measure metabolites within the muscle and measure mitochondrial related parameters. So with MRS scans, things like that, they can be measured differently. But when the muscle is obtained from a muscle biopsy from a participant, and then we can only work with what we have experimentally. But we could play around with the oxygen concentration, because that's also a big factor, because this is room air, and I think that's the biggest factor when we model the differences in our muscle, is that that's not the same oxygen tension within our muscle compared to what we measure experimentally. So I think that's a big factor that could be changed.
Okay, one final question. Your last study, where you were investigating the potential role of decreased ADP sensitivity in the mitochondria for resynthesis of glycogen after exercise, I was a bit perplexed why you might have expected anything, given how slow, relative to exercise, we resynthesize glycogen.
We saw in a different study in mice where we provided insulin injections to mice that resulted in an increase in ADP sensitivity. So then our rationale there was that decreases the cytosolic ADP concentrations, brings more ADP into the mitochondria, and maybe that's a mechanism that's more related to the anabolic nature, so like the storage nature that occurs with insulin. So our thought process was that maybe that's involved in the storage of glycogen, because there's also an insulin response when carbohydrates are ingested. So then the rationale from that was from some other work we had done, but then in the case of the in vivo, more like practical situation in humans with carbohydrate ingestion, then it didn't seem to be the case, because of course there's so much other regulation on glycogen synthase that exists.
Okay, very good, Ms. Candidate. I'll hand it back to the pro-rector.
Thank you. The opposition will be continued by Professor Witkamp, Professor Nutritional Biology at the Wageningen University here in the Netherlands. Professor Witkamp was also a member of the assessment committee.
Thank you very much, Mr. Pro-Rector. Respected candidate, first of all, I would also like to congratulate you with the completion of your impressive work, and I would also like to congratulate both teams here in Maastricht and also the one in Canada. It's clear there is still a lot to discover about mitochondria, but you really added to this field. I would like to start with the findings on nitrate, which are kind of spectacular, I would say, and that was about chapter 3 and 4. But I'm also a little bit confused sometimes, because it seems that nitrate has a lot of positive effects. So we should eat more vegetables, and we are actually, athletes already know that we should take beetroot. And at the same time, there may be some disadvantages of nitrate consumptions. And the first one, I've always been taught that the combination of protein, the potential carcinogenity. So what would you recommend regarding nitrate consumption?
Dear highly esteemed opponent, thank you for your question. That is a bit some debate about the carcinogenic effects of nitrate, but those studies were all used in mice with extremely high doses that are not obtainable in humans. And generally the negative effects of nitrate are associated with nitrosamines and formation of other compounds like that, which are in processed meat, things like that. And those compounds interact within the food of the processed meat or something, which is different than a nitrate in a green leafy vegetable or sodium nitrate supplementation.
So you really say that's something of the past. Well, when I studied that was something to concern me, you say, okay, we can forget that. The other thing is tolerance development of nitrate. How would you, let's say, value that? How would you evaluate that in terms of consumption?
Well, it's known that ingestion of a single dose of nitrate increases plasma nitrite concentrations rapidly within 2 hours, and that occurs even with sequential ingestion of nitrate. So even after a chronic supplementation period, there still is the rise in nitrate and nitrite. So then that can be translated to assuming there is still a biological effect of it as well.
Yeah. And your results seem to, and it has already been addressed, that your results are typically for nitrate, so not for nitrate. But can they really be disentangled? And also considering the difference between mice and humans there. So when you look at the overall effect of consuming nitrate, what are the effects of nitrate? And what are the nitrite, I would say, the NO2? And what are the typical nitrate effects? And can we really disentangle them?
We often think the outcomes are due to nitric oxide, so the end result of the pathway nitrate, nitrite, nitric oxide. The nitrite levels are many-fold, 100-fold or so lower than nitrate. So the majority of what we ingest is nitrite. When we measure in the body, we do measure nitrate plus nitrite NOx concentrations. They're kind of combined. So I guess disentangling which is nitrate and which is nitrite is quite hard to do, but they all move through the same pathway to produce nitric oxide, which can have biological effects.
So you think it's at the end is nitric oxide what's doing the job. So coming back to the effects of the microbiota, well, it has been addressed also by Professor Goossens, that it could be just an effect on the microbiota. But at the same time, there are studies being done in germ-free mice where nitrate does not have that much of an effect because apparently the conversion to nitrite is not taking place. So what are your thoughts about that?
The gut microbiota and the relationship with nitrate kind of goes back and forth because the microbiome, especially the oral microbiome in the mouth, is important for converting nitrate to nitrite and ultimately nitric oxide. So without the bacteria, then there wouldn't be the same conversion of nitrate to nitrite. To nitric oxide, and then the biological effects would be absent as well.
So what would it mean for your results then? What would you see on mitochondria?
Then
you still think you would see the effect by nitrate then, or?
I would expect to not see an effect in the germ-free mice because if the nitrate isn't converted in the body, then we would expect to not have a downstream effect on our tissues as well.
Yeah, and then going back to humans, in the human gut, it's apparently not being, let's say, metabolized in the intestine, it's being done in the saliva. Mm-hmm. So how would your result then translate back to humans?
There is some evidence in humans that mouthwash can prevent the conversion of nitrate to nitric oxide. So something like that in humans would maybe then diminish some of the effects that we observe with nitrate. It's obviously harder to control for more other external factors in humans. But there always is some gut microbiome or some microbiome in humans to kind of facilitate that conversion.
Yeah. And a kind of philosophical question when it comes to development, the future development in terms of nitrate, in terms maybe of pharmacological development. What is your view on that? Are we going to see more, because we already have this organonitrates used against hypertension? Mm-hmm. Do you see more development there? Should we increase consumption, or you see new molecules being developed?
Yep, so sodium nitrite, NaNO3, is used often as well. The downside is that it's attached to a sodium molecule, so there could be some potential less beneficial effects given the sodium can contribute to hypertension. Of course, with NaNO3, there still are beneficial effects on blood pressure and whatnot. But in terms of a health perspective, the consumption of green leafy vegetables, beets, and spinach, things like that, would be the most preferred way of supplementing with nitrate, since it has the nitrate as well as other more beneficial aspects of the vegetables.
Yeah, okay. But there is a limit to that. So you not think about new molecules being developed, supplements or something, concentrated forms of nitrate.
Concentrated forms in terms of right now, concentrated beetroot juice, or
but not more. You keep to the natural way.
The food way is, I would say, more beneficial than pharmacological.
Yeah, and then to switch to the ketones, which is also interesting what you found. First, a very general question. If I look at your results, also regarding the high fat on the one side and the lack of effect of ketones as a energy supply, at least in the mitose, what's your opinion about the ketogenic diet? Also very popular, of course.
Yep, I think the ketogenic diet would kind of downregulate PDH and prevent carbohydrate utilization. So I see a little nice there. And in terms of exercise, that's our main currency is carbohydrate utilization. So in exercise context, it would be a negative effect for preventing us from using the energy source that we
well, it's also popular for health reasons. All kind of positive effects are being attributed to that's a ketogenic diet. Would you recommend that?
I would not recommend it, just because the high fat intake, as we've shown, has many negative effects on the whole body outcomes that we measure. So I think in that sense, carbohydrates are still an important part to be a factor of the diet to be ingested.
Yeah, at the same time, you talk about evolution. We are adapted to ketogenic circumstances, of course. So, well, it's in the terms of the feast and famine theory. So there were, let's say, periods where we did not have enough carbohydrates, and then we were adapted to ketogenic diets. And then coming back to the administration of external ketones.
Can we continue, please?
Very short?
Is it a very short question
Okay.
then? Okay, and short
It's
answer.
still being done, also against heart failure. And that seems to be kind of opposed to what you found. So what's your opinion about that?
Yep, I think in a clinical setting there's some potential for ketones. It's been shown that during heart failure situations, fat oxidation is decreased in the heart, which is a negative change, and ketones could have some potential there to provide an alternative fuel for the heart. But that's a very different context than the use of ketones in skeletal muscle in different situations.
Okay, thank you very much. I'm very happy with the answers. Give the word back to the prorector.
Thank you. The opposition will be continued by Dr. Miller, Associate Professor of Cardiovascular Physiology at the University of Guelph. Dr. Miller was also member of the assessment committee, and also for you, thank you for coming over for this academic ceremony.
Thank you, Mr. Prorector. Dear Mrs. Candidate, I would like to extend congratulations to you as well. It's a really tour de force of a thesis, and I hope you were able to read some of the comments that many of the assessors provided to you, because I think it really highlights the breadth of work that you've been able to do in this thesis and the different areas and approaches that you've been able to learn and manage as you've answered some, next step questions to figure things out. Obviously, I think you know what direction I will take for my questions, so I'm going to be focusing primarily on Chapter 4, and I want us to think about, in my world, as a cardiovascular physiologist, and I think about giving nitrate, dietary nitrate, the most consistent evidence would be to have a hypotensive effect. In your study, though, you actually see that blood pressure, mean arterial pressure went up. And so I'm just wondering if you can comment on that and clarify why you think that occurred.
Dear esteemed opponent, thank you for your question. Indeed, we found we did not see a decrease in blood pressure, which we found interesting as well. Of course, we just measured mean arterial pressure, and we could have maybe seen something different if we measured it at the aorta, if we had measured in the pulmonary trunk, where we also saw changes in diameter, things like that, or if we measured more peripherally. But I think it has been shown in humans quite often that nitrate can lower blood pressure even in obese situations, which is what our motto was. So it intrigued us as well, and we did not have an explanation for why.
Okay, so you have some cardiac function data, you have some blood pressure data, you look at blood pressures going up, cardiac output is not different between your low-fat diet and your high-fat diet plus nitrate. What does that suggest? What's driving that pressure response?
So the cardiac output was not different between our low-fat and our high-fat. And stroke volume was maintained and end-diastolic volume was maintained. So then that shows that there's still the same filling to the heart. So maybe the higher blood pressure then was promoting kind of like the filling of the venous return to the heart to fill the heart to keep the end-diastolic volume maintained.
So I was thinking more like Ohm's law. If you remember back a long time ago, probably when you took exercise physiology, we're thinking about if the cardiac output is staying the same and the pressure response is higher, that it has to be a total peripheral resistance response. There has to be some vasoconstriction somewhere in that system. And so just trying to understand where that could be, given that nitric oxide is a dilator. In my world as well, nitric oxide is a central sympatholytic, which again would be lowering resistance. So somewhere in that circuit, in the peripheral circuit, you're having some constriction. And so I think kind of understanding that because in the long term, I understand in the study you've demonstrated the ability to ameliorate those cardiac function deficits in the 8-week window. But what do you think chronically, right? So if you take someone and say, you know, for the next 10 years, 20 years of your life, you're going to consume nitrate in a dietary form, maybe, to be able to achieve these benefits. Do you think that there's a concern, or do you think that it's not as relevant as a translation from the model to a human sense?
I guess in our model, we also use NaNO3 as our nitrate form. And our control mice, we dissolved it in the water. The control mice just consume normal water. So the extra sodium in the drinking water could have had an effect that if we had controlled for it with an NaCl in the same concentration dissolved in the drinking water, maybe that would have been a more fair comparison between the effects on blood pressure. So maybe if it was through like vegetables or something like that, then we maybe would have not seen that response that we did with nitrate.
To get that dose, you would have to consume a very large amount of those vegetables, would you not?
So the 4 millimolar that we use equates to about 0.2 millimole per kg, and in a human then, say an 80 kg human, that's about 16 millimolar, which is achievable through beetroot juice and nitrate and foods. It's on the higher end, but still feasible with a high vegetable ingestion.
Did you consider plasma volume, right? So you have a lot of changes in end-diastolic volume, and from some of the supplemental data, it looked like maybe water intake was a little bit lower in the high-fat diet groups. Was that considered given some of the cardiac function data? Because most of those are going to be preload dependent.
Yep, that is possible as well if we had a change in plasma volume, especially if more water was ingested or more that NaNO3 was dissolved in the water, so had more osmolarity in the plasma and more response to increased plasma volume. That would explain why blood pressure was not lower if there was an increase in plasma volume, and then also could explain why there was more filling towards the heart as well.
Okay. I have one, okay. One short question. It would be do you think that there's a difference in your cardiac function data between the fecal transplant from when they had the high-fat diet plus nitrate versus the low-fat diet alone? So in the supplement, you showed those two comparisons. Do you think that they're operating through a similar mechanism or different?
I think it would be through the same mechanism of the changes in the gut microbiome, because the microbiome composition of the low-fat and the high-fat nitrate were quite similar, and that was what we provided the FMT with. So I think it was all due to the changes in the gut microbiome that then is having an effect on the heart.
Okay, thank you, Mr. Prorector.
The opposition will be continued by Professor Galloway, Professor of Exercise Physiology at the University of Stirling in Scotland. Professor Galloway was also member of the assessment committee, and also thank you for coming over.
Thank you, Mr. Prorector, and Ms. Candidate. I would also like to echo everyone else's congratulations to you on the excellent body of work that you put together in this thesis. And the range of techniques is amazing to undertake within this period of study. I have some more general questions as well for you, and I'd like to start by asking you about species differences. And this question popped up because early in your thesis you were referring to some animal work around high-fat overfeeding stimulating mitochondrial biogenesis. But I think you're probably aware in human studies, when we feed high-fat, hyper-energetic diets over 6 days or up to 8 weeks, we don't see the same increased expansion in mitochondrial volume. So why do you think there's that species difference occurring?
Dear highly esteemed opponent, thank you for your question. I think a major difference between the rodent models and humans is exercise, physical activity. So at least in chapter 4 in our high-fat diet-fed mice, we didn't see any decrease in activity between the high-fat and the low-fat mice. But that's not the case in humans. So on a high-fat diet, then generally in situations of obesity in humans, then you also have a reduction in physical activity, which can influence mitochondria. So in that sense, if there was less physical activity, then that would be a large stimulus to cause the reductions in mitochondrial biogenesis.
Okay, so the high-fat overfeeding stimulates activity in the mouse model. Is that what you suggest?
It didn't stimulate activity, but there was no difference. So, okay. So then that's why it could be different than from a human model.
Okay, great. And also, I think you argue very nicely for the use of your isolated mitochondrial methodologies. I just wondered, being a bit old school, whether is there still a place for measuring maximal activity of mitochondrial enzymes, maybe to get a handle on mitochondrial volume changes as well as functional changes that you're measuring?
Mm-hmm. So measuring maximal capacity still is important, especially with interventions that increase or decrease capacity, so exercise training and mobilization. Clinical situations like that where there are known changes in maximal capacity. But in certain diseases, then we see that the submaximal, it's what's regulated more. So they're both important to measure and to interpret together. So I think you need one to interpret the other one properly.
Okay. No, that's great. I'm happy with that answer. If I move on, just again, related to metabolic disease and insulin resistance, there's a whole body of older literature, I guess, looking at in high-fat overfeeding situations that the accumulation of lipid intermediates in tissues because of mitochondrial overload essentially leads to impaired insulin action and impaired glucose transport. What's your thinking of that, how that model has changed in recent years, and where do ROS fit into that model?
I think reactive lipid species accumulation in peripheral tissues, especially muscle, is a big factor that causes insulin resistance. ROS can be increased due to the high lipid environment. And the high lipid environment also decreases the transport of ADP into the mitochondria due to impairments in ANT. So less ADP being moved into the mitochondria then can increase the propensity for ROS. So the effect of the lipids on ADP transport then has a consequential effect on elevating ROS. And that in turn has many effects on insulin signaling in the cytosol as well as other inflammatory cascades.
Do you know specifically how ROS might act on insulin signaling? Is there a potential mechanism there, and also potentially on GLUT4 translocation itself?
Yep, so it can influence IRS1, which is involved in insulin signaling at the plasma membrane, and can also have effects on that, then in turn has effects on GLUT4 translocation to the plasma membrane for less glucose uptake.
Okay, no, thanks for that. Am I allowed to ask a slightly different question, if that's okay? Yeah. I just want to switch to your ketone chapter and just ask about what sort of level of ketone increase would be observed in your model where in humans they were overnight fasted, they did a glycogen depleting exercise and then a 12-hour subsequent fast. Were ketones being elevated as much as you would see with supplemental ketones that cyclists, as you alluded to, are taking in their drinks?
The increase is much lower after an overnight fast than will be observed with supplemental intake. So it goes up to about 1 millimolar of ketones after an overnight fast. And I think with supplements, you could easily get to 3 or 4 or 5. So the increase in ketones in the body with a ketogenic diet or with fasting is much slower over time, whereas with supplementation, then it's a rapid increase. And that's kind of the different metabolic situations that you have in those situations.
So would you suggest that supplementation is a suitable approach in an athlete situation?
I would suggest that neither of them are effective approaches in athlete situations, but the difference with the supplementation is that it does not require a low carbohydrate situation. So it can be normal high carbohydrate availability and also on top of that ketones. Whereas inducing ketosis with a ketogenic diet requires very low carbohydrate availability, which in itself is a negative consequences on performance.
Okay, thank you very much. I'm very happy with the questions. I hand back to Pro-Rector.
Thank you. The opposition will be continued by Professor Faber, Professor of Neuromuscular Disorders at this university. Professor Faber is also the Secretary of the Degree Committee.
Thank you. Dear candidate, first of all, I would also like to congratulate you with the results of your efforts and with a very beautiful thesis. I would also like to congratulate your supervisors. In chapter 3, and that has been discussed already a bit, you studied adipose inflammation, mitochondrial dysfunction, and gut dysbiosis. And then you conclude that especially the high-fat diet contributes to the problems that we see, more than obesity on its own. That's true, isn't it? Yeah. You have also shown this in the graph on page 24, which I like very much. But what would be the translation of these results to our daily clinical practice? Can I translate this to patients or to humans? And what should I do?
Dear highly esteemed opponent, thank you for your question. I think in translating to humans, then the key takeaway here is that diets should not be composed of a very high amount of fat, because that can have negative effects on mitochondria, whole body outcomes. And even if it's not necessarily caloric overconsumption, even a high fat intake can have still the negative effects. So in terms of a clinical perspective, then of course we want to ensure adequate protein is consumed, but then also something to keep in mind that its fat percentage is not overtly high as well.
And does the type of fat make any difference?
The type of fat definitely can be involved. So for example, the high-fat diet we use in mice is made out of essentially large, so saturated fatty acids. There's some evidence that omega-3 fatty acids as part of a high-fat diet are beneficial. So the type of fat can definitely influence the outcomes that we see.
Yeah, so we should use olive oil, fish, or whatever, and not the, well, cookies and, yes. I can understand that. Oh, I'm sorry. And then in chapter 4 and 5, and one of the other opponents already asked a little bit about that. But what should be advised to increase daily nitrate intake and to avoid negative effects? So is only beet juice enough, or should we do more?
So beetroot juice, also foods like arugula, spinach, things like that, even making like a green smoothie, for example, where it can be a bit easier to get the amount of vegetables consumed. But I would advise natural sources in vegetables as opposed to a pharmacological approach.
Yeah, and if you have too much nitrate, what would be the negative consequences?
There could be some negative effects over a long-term duration.
You can finish your answer.
Thank you. It's not yet fully investigated the effects of, for example, years of chronic high nitrate consumption. There could be some, could be a negative effect, but at least in what we see, there is consistent beneficial effects of nitrate in a longer duration.
Thank you very much.
Petrick, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and now return to this room.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
side. 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Your mind, years can't fly by now, don't waste all your time, cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time, cause I'll go the extra mile.
Dear Petrick, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Van Loon is authorized to confirm upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Heather Lauren Petrick, cum laude, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate, signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Luke's asked me to speak on behalf of both of us. So thank you very much for coming today, everybody in the audience, and to the examining committee. We almost didn't have one examining committee, so a big Herculean effort to Dr. Whitman, who somehow managed to shave 15 minutes on his commute and show up right at about 3:58, so that was fantastic. Also, 3 people that have flown in, Professor Stuart Galloway, Professor Lawrence Spreet, and Dr. Phil Miller, so thank you very much for coming. I know it's particularly special for Heather to have you guys here. And I think it's a testament that we've got so many people in the audience that have come from the UK, Germany, Canada, Sweden. I think that speaks to the impact that you've had in 2 different labs over a very long time. So I'm supposed to give a bit of context in terms of how we ended up here with a double degree and maybe your experience along the way. I think Heather also wants everybody to recognize that the 3 people that flew in are giving talks tomorrow, and so if you have really difficult questions that she would like you to come tomorrow to ask those 3 gentlemen, that would be fantastic. But I was thinking about how we ended up with this double degree. I have lots of stories that I could share. Heather joined my lab as a co-supervised master's student starting in 2017. Then I could talk about your challenges of trying to balance 2 different professors with your running career. I could talk about why lab mates nicknamed you Poop Girl. I could try to describe the impact that you had on my group as we navigated COVID, and it was just the 2 of us literally trying to keep our research program going forward. Then I started to think about ACSM in 2019, talking about ketones with Louise Burke and Luke having this brilliant idea to do a double degree and trying to come to the Netherlands to advance your research and to learn how to do human clinical trials. But I kept coming back to my very first experience, which was not actually Heather speaking, but Heather writing. And in 2016, she wrote a midterm in my course, and it was about 6 weeks in, and I'd never talked to her. She came to class, never said anything. Just reading long answer question after long answer question, giving full grades, full grades, full grades, going, who is this Heather Petrick person? And it really highlighted to me the innate intelligence that she had, the academic prowess. And I started to think, this is somebody that I would want in my lab. So that's actually why I targeted Heather to recruit her as a master's student, was from that experience. It was very prophetic. I think that's probably why Luke, when we were talking about ketones, also wanted to work with you and to initiate this collaborative co-tutel. And that has served you well. I think last I checked, you have 34 publications, 12 or 14 first author. You have secured the highest funding in Canada for your master's, the highest funding in Canada for your PhD. There's one Brock doctoral scholarship that we give to incoming PhD students at the University of Guelph. Heather was the recipient. We give one Governor General to the highest convocating PhD student, and she is still in the running, so she's got a 1 in 7 chance of winning that for the June competition. She's won early career researcher awards when she spent time in Maastricht, going to ECSS, so it serves you very well. But that midterm was prophetic in another way, because on the back page was a handwritten note from Heather saying, "I apologize for my messy handwriting. I was hit on my bike ride on the way to university." And I don't think I'm supposed to do this, but I need to apologize to Luke because I don't think he quite grasped what he was getting when he offered a coat to tell, because he didn't realize the injury-prone individual that he was getting and probably the emotional turmoil that she has exposed him to over the last 5 years. I think your parents know, because I'm pretty sure they bought you a bike trainer in Guelph to try to keep you off the road, whether it's running and rolling your ankle. I can talk about rolled ankles running in Dublin. I can talk about numerous times you've fallen off your bike. And Luke, has lots of stories about meeting you in the emergency room on his vacation in Spain for the same reasons. So while I'm very happy that you've graduated today, I'm probably more happy that I feel no more responsibility for you, and I can actually hand the baton over to Luke, and I take no responsibility for what you do from here on out, and that is entirely going to be on him as you continue your scientific journey. But obviously, we are here to celebrate you. We're here to celebrate the success that you've had, a great thesis, and you heard it from everybody today. It is using Dr. Miller's verbiage, tour de force, in terms of the science that you've done. You should be very proud of it. I know we are very proud of the work that you've put in, and we're excited to see where life takes you. So congratulations.
Dear Dr. Petrick, also on behalf of the Maastricht University and the Board of Deans, it's a pleasure for me to congratulate you with this double degree, and particularly with the predicate cum laude. That's quite exceptional and illustrates, in fact, the quality of your scientific work. Referring to one of your propositions, Proposition 10, I wish you a lot of positive energy in your future scientific career, and I will wish you all the best in the future. Saying that, I will close this ceremony. I will ask the audience to leave now this auditorium, and in the meantime, and to go to the reception room, and in the meantime, we will make a picture with the new doctor and the family on the stairs in the central hall.